It's working?
Yes.
Okay. Shall I start? Yeah, thank you. Thank you. My name is Ayan Rajaudhuri. I'm a postdoc with Professor Madan Rao at NCBS. So actually Madan was supposed to give this talk, but he was not feeling well, so he just sent me here. So this is about a few experiments we observe in these actomyosin systems. So this is a cell just plated on a substrate, and it is crawling. And here, what you see in red is actin proteins, and the green parts are microtubules. So as you see, as it crawls, so they are forming these sharp lines. Okay, so these are called stress fibers in biology, and the whole point of this talk is writing down a continuum theory of emergence of the stress fibers. So it's a mixture of this actin and microtubule particles and also myosin proteins. So there is a actin fiber in the background, and these myosins, these are motor proteins. So they like sit on this actin like a fiber, like a fibrous meshwork, and they pull on this fibrous meshwork, and this myosin can bind and unbind from this meshwork, and as a result of this, so this like active dynamics, they sort of form these sharp lines, and we look at these lines as okay. So these myosins, as they pull on the actin like meshwork, they are also like correlates with the force in the meshwork. So these sharp lines also are the lines where the force is the maximum. So these are the stress. So you can say that these are the like lines where stress is concentrated in the material. So the whole like the like my work is about how in such active systems stress gets concentrated along these lines, like as this system is moving. So these are other experiments. So people have like plated a cell on a, like this tripod shape substrate, and here also like as you turn on the myosin activity, they start producing these lines, and it's a dynamic like it's very dynamic, and stress is concentrated on this like sharp fiber kind of thing. So this is just a schematic of Manuel Theory's group paper. So so this background fiber of actin, it is actually turning over. So actin can like polymerize, depolymerize, but like in this in these experiments, they have stopped the the actin polymerization. So so our work is motivated from these experiments, where you can consider okay, so in general this meshwork kind of thing, so it's a fluid meshwork, because because the constituent is actually binding, unbinding. But for our our theory, we will restrict to a elastic system. So this meshwork is a connected meshwork, and the activity the activity is coming from the myosin only. So no actin turnover is here. So these are some other experiments where so you start with stress fibers, and you wash out myosin, and the stress fiber vanishes. And then again, when you reactivate myosin, the stress fibers comes back again. So these are just some experimental motivations. So the work is about how you get stress concentrations in such active elastic systems. So why elastic? Because our our system, there is no turnover of the constituents. So it's a steady meshwork, and on top of which this clips-like thing, these myosins, these are like binding and unbinding, and they are like pulling and pushing. So this is the system. Okay, so this is just a comparison. So with granular media, so in dry granular media, you see you also see stress concentrations, but these are compressive force chains. Whereas in our case, these are the same force chains, but these are tensile force chains. So they are under tension, and like compared to granular media, which are under compression. So, so this theory that I am going to present, you can like think of it as also a continuum theory of like granular media force chains, which is right now absent. So this is just like it's not so in granular media, this the like how you get force chains depends on what kind of forces you apply. So like there are shear jam systems, there are like this pressure, you put isotropic pressure. So it's like they depends how you get force chains, but it's sort of like an understanding from a continuum theory perspective, like at what situations you may get stress concentrations in a continuum theory. Because in elasticity, usually the stresses are diffusive. So, so this talk is about how you get out of that diffusive stress situation. So this is our system. So, so as I said, it's a meshwork of these actin fibers, and on top of it, these myosin motors, like these small things, they are binding and unbinding, and they binds with certain rate and unbinds with certain rate. And in case of myosin, it has been found that this either it could be binding or unbinding rate. They could depend on the local state of the meshwork. So in this work, we are just assuming that the unbinding rate is strain dependent. So so there are two components of this, role of activity here. So activity can be in terms of force sensing, like through this dependence on strain, and this is the general notion of activity, that it generates force. So these are the two aspects of activity in this system, and these two aspects, they are actually independent of each other. So this force generation comes from ATP consumption of by the myosin motors, and this force sensing, it comes from some other signaling pathways, and these two aspects are unrelated to each other. And this is the origin of what we see as what we will later see, that such systems are called non-reciprocal systems. Maybe you might have heard this term yesterday from Vijay's talk. So this this is the origin of non-reciprocity in this system. So this is just the schematic, and the what we do here in this work. So this is like a microscopic picture. So we will write continuum theory of such an active meshwork, and so we will write basically differential equations for the displacement vector u here, the density of the actin meshwork, and the density of the myosin motors. And in actual systems, so this subscript I, so this is rho 1, rho 2, rho 3. So in actual cell, there are many species of myosin motors, and they have different turnover rates and different contractile activity strength. So our theory, we have written down the theory for two species of myosins, but it can work for just one species of several species as well. So what is happening here is how we are getting these force chains. So this is the theoretical picture. So we will write down the equations for all these variables, and we will see that if you start with a homogeneous density of myosin, like a random mixture of myosin on this meshwork background, due to this activity and this turnover, so this material renewal, we will see what it is actually, but this material renewal is actually this force sensing aspect. So due to this activity and, sorry, yeah. This is the density of this myosin motors, and this is no, sorry, this is actin. This rho A is actin density. Yeah. So rho A and U, they are describing the elastic material actually. So it's the density of the elastic material, and U is the deformation of that material. And Yeah. So they are related. They are related. They will be related. So you can write down different, like you can. I'll come to it in a while. Okay, so we will write down, okay, so where I was, yeah. So due to activity and this material and this force sensing aspect, we will see that these myosin motors that are homogeneously distributed sort of on the material, they will undergo a phase segregation. So they will form like myosin rich and myosin poor domains. And the crucial thing is these myosin motors, they don't have any attractive interaction among them. So this phase segregation that we will see is purely coming from the elastic interaction that they are having, the background interaction. So that is some new kind of phase segregation, and this phase segregation will also allow some to trigger some floppy modes in the material. So this is an elastic material, and these myosins are pulling and pushing, and what will happen is it will change the elastic moduli of the material. So we will write down, like we can write down an effective elastic moduli for this material, which will depend on the myosin density, and we will see that some of the effective moduli can go to 0, and it will trigger some soft deformation modes. And the physics is this accumulation of these deformation modes are what we see as stress fibers, like just like these wrinkle lines, So this fabric has a soft mode, like it fails under compression. And these wrinkle lines that you see on the cloth, they are sort of the stress fibers we see, and they are the lines where stress is concentrated on these drapes, like in any soft material we see. So that is the main message of this whole talk. So I'll just quickly go to the equations. So so we will write down the governing equations of our system. So this is an overdamped system. So usually like divergence of stress, and we will equate it with the inertial term. But this has a body force, which is, so this actin meshwork is immersed in a cytosol, which is a fluid, and it is so it is experiencing this friction with respect to this fluid. So this is an overdamped elastic system, and the total stress has these three components. So this is the passive part, and this is the active part, and this active part, this active stress will depend on the local actin density and local myosin density. So we are taking a special form where it depends linearly on the myosin density. And the elastic part has this usual, like it comes from a free energy, and free energy, like it has this general form. So it has, like we are assuming that it is an isotropic meshwork. So it has isotropic elasticity with two moduli. And okay, so this is the part where usually if you have large density variation, so there people write this kind of terms. Okay, so this is from this is not in standard elasticity we write this density variations, but this we write where you can have diffusion driven density variation, large density variation. So there we kind of write these terms, but here in this work we will not write separate equations for this actin density. So we will simply assume that this density variation is left to the strain. So if you are so we will not write separate equations for the actin dynamics. So just this. So for the background solid, there is just this one equation, and this is the equation for the bound myosin motors. So the myosin motors, they are bound on the meshwork. So yeah, so they can like advect with the meshwork velocity, and there is a turnover term. So it can bind with this rate, and it can unbind with this rate, which depends on the strain, local strain of the meshwork. And So this dependence, this is a standard form. This is called Bell's form. So it's taken as an exponential function, and this coefficient alpha you see. So the sign of this alpha, so it dictates whether the myosin bond, like bond with the fabric, it's of catch type or slip type. So catch type is like you are pulling the background actin, and more myosin is binding onto the system, and the opposite is called the slip bond. So so this is the these are our equations, and this dissipative stress is just like for this talk. This is just for regularization. You can see like we will be solving these equations, and this will help like regularize the dynamics mathematically. So so in our case, we have taken two myosin species. So we will see that there will be a segregation, so ρ1 and ρ2, and we will recast these equations in terms of the average density and the relative density of the species, and this will serve. Yeah.
What is the difference between epsilon P del epsilon?
Yeah. So this is the trace of the stress of the strain tensor, and this is the deviatoric part. There is supposed to be an equation, but rho a dot doesn't evolve separately from strain, so we won't need an equation for that. So so we needed an equation if there is actin polymerization in our system. So we are not considering polymerization or depolymerization of actin right now in this work. So yeah, so now coming to this, yeah, so just briefly, so this active stress, so it can be a general function. So this is just this is just a simplifying assumption, and this function chi, this is like experimentally people have measured it. So this is like a sigmoidal function. So it rises up and then saturates, and so what we will do to get an exact form of the active stresses, we will like Taylor expand this function in as a small variation. So this is the actin density. So we will Taylor expand, and we will keep up to certain terms, and we will substitute this form in that Taylor expansion. So ultimately what we will get is so this is the hydrodynamic final equations. These are all non-dimensionalized. So this is the deformation equation, and these are the ρ1 and ρ2 equation written in terms of average and relative densities, and so this has now become the total stress. So this is elastic stress plus the active part, and so they have different contractile activities and different turnover rates, and in mammalian cells, people have found actually 3 species of myosins. They are called like non-muscle cells, myosin 2A, 2B, 2C, and they. I don't need, but yeah. So this whole theory works with one myosin species also. So in that case, we will write down. So just there will be one equation for myosin, and still you will see a phase segregation there. So so this is the whole, the expression of the full stress. So what I have what you have to do is like Taylor expand this and substitute this expression and keep certain terms, and how many terms? Like it's just a Landau form. So here I have kept only the cubic term of the stress. So what is the outcome of this? The outcome is that the total stress has now a pre-stress, which is purely active. So this depends on the myosin densities, and activity has also renormalized the effective moduli of the system. So this was the passive, this was the modulus of the just the passive meshwork, and now due to the presence of the myosin motors, you get a new bulk modulus, and these two B2 and B3, and it goes on. So they are all active, like activity introduced, higher order bulk modulus. And you can also assume that okay, so mycins are actually, like usually assume that they are, so there is a homogeneous distribution and they are isotropically pulling on the meshwork, but they can also put shear forces, shear active forces. So you can introduce shear active terms, and in that case you will get other moduli, other shear moduli. But here, like, we are only allowing isotropic like activity, so that's why we are getting this isotropic renormalization, like of the bulk modulus only. So what will happen? So you see that, like, in regions where myosin is high, the effective bulk modulus is like it will reduce, and so it can actually take the material to regimes where the bulk modulus can approach 0 or even negative values. And similarly, yeah, so what I was just telling, you can do the same thing with shear modulus also. So, so what this effectively is doing, this myosin species, they are changing your background material itself. So you are getting a new material with new moduli. So this is just a small detour. So so this is just a phase diagram of of an isotropic solid. So we have bulk modulus and shear modulus, two moduli, and so this regime where these modulus are positive, so your elastic energy has this parabolic shape, paraboloid shape. But when you make certain moduli 0, for example, here we are making bulk modulus 0 on this line. So here you get this floppy directions. So here this deformation. So you are like compressing, and this compression is floppy, and this is the expression here of the energy. And similarly, your shear modulus can become 0, and it can the shear direction can become floppy. So this is and these two regimes are correspond to the classical spinodals. So what we know as spinodals, like you have this this energy landscape, and where the energy landscape loses its convexity. So this is the classical spinodal. But in elasticity, so this is the strain, right? So so this landscape is like your axis are strain axis. But interestingly, in elasticity, actually, your primary variable is not strain, it's a displacement. So my order parameter is a gradient of another variable. So because of this fact, in elasticity, you get certain another notion of spinodal itself. So it's not with respect to your like primary variable, like where you plot your energy, but it's another variable like displacement. So so in elasticity, you get some special spinodals which are not there in fluid-like systems. So those we have termed like elastic spinodals, and where they come from just a linear stability analysis of your elasticity equations. So this is just divergence of stress equal to 0, static equilibrium equation, and you write your like you put your expression of strain in terms of the displacement, and you consider the stability of that equation with respect to small perturbations in displacement. And then you, like, it's the standard thing, you go to this Fourier space and look at the eigenvalues. And the eigenvalues here, instead of like shear modulus and bulk modulus, so this curious combination appears, this B plus mu. So this is two-dimensional elasticity. So instead of these two lines, so like these were my classical spinodal lines. In elasticity, you get the other, like a new kind of spinodal line, where so this is the usual fluid where shear modulus vanishes, and this is like a crazy material where bulk modulus can be negative. Okay, and this is actually so the point is when my material, so this active material, actually this myosin motors, they are pushing my material to these lines, either this or this. And when you reach these lines, what will happen is this PD, it's a elliptic PD. So in elasticity, when you put a force here, so this force is getting diffused throughout the system. But when you will reach these lines through activity, the nature of the PD will change and it will become parabolic. And the stresses will propagate on the characteristic lines of that parabolic PD. And those force chains that so the whole point is that the material is, activity is taking the material to this line, and the stress fibers are actually like these characteristic lines of the system that we see. So this is just the same thing. So we are starting from normal elliptic regime of elasticity, where the deformation is smooth. But when you hit these lines, this elastic spinodal lines, you get, in case of where shear modulus vanishes, you get this slipping, the shear bands. And when this combination vanishes, you get this compression and expansion bands. And when you hit this bulk modulus equal to 0 line, nothing happens actually for infinite systems. But for finite systems, you get something called surface instability. So our focus is on this material, and this has been historically called an elastic ether. So this material was proposed as the first theory of light before Maxwell's electromagnetism theory arrived, and people were calling it as a mechanical theory of light. So people like Cauchy, Lord Kelvin, they proposed this theory, and this is a nice book on the history of this interesting material. And you are seeing that, so the energy landscape, so this has a single minima. When you hit the classical spinodals, you get this floppy directions. But for ether, it is actually, it has, the curvature is negative here. So, and you get these degenerate lines. So this is the energy landscape. And these degenerate lines, you have seen, you are seeing it as these two branches. And these two branches will correspond to either compression band or expansion band. So that is not important. And when you plot the Green's function for elasticity, so Green's functions are solution to a point forces. So here we are applying certain point force systems, and when you are in this regime, so it's a smooth Green's function. But when you hit this spinodal regimes, so in case of fluid, you get this shear bands, and in case when you hit this line here, so you get this. So this is the plot of the displacement field. So you get this compression and expansion bands. So this is just the elastic Green's function where you have reached these lines. So this was just a detour, and okay. So in all these calculations, the whole material has been taken to this regime. So my whole system has reached this either two kinds of spinodals. But going back to our original system, okay, there is another slide. Okay, so what is this? So this is, I am putting concentrated forces at these four centers. And when you have hit the ether line, and when you have actually crossed that line, so B plus mu is actually negative here. And this is the energy landscape, and you have put some small nonlinearities in the material so that your energy, so that you get finite minima. And what I have plotted, this dotted line is the classical spinodal where it has lost convexity, this reference state. And this dark line you see, this is the elastic spinodal lines. So in regimes where the strain is inside this region, you will get this force chains. So these are force chains, tensile force chains, and this length scale that you are saying, of course elasticity has no length scale to simulate this, we have put certain gradient elasticity, certain strain gradient term in our equations. So, so in usual, like if you are in the usual regime, you put these forces and it will diffuse. But here, like you get chains, like this chains will connect these lines, this force application centers here. And this material is a homogeneous elastic material where the whole material has these properties where B plus mu is 0. But for our actomyosin system, our actomyosin system has like it's a heterogeneous material because the moduli is not same everywhere. So, so this is just a 1D simulation. So we are starting from a homogeneous dense, some random some Gaussian noise kind of myosin distribution, and activity is on, and you are getting a phase segregation, and it will like what you are seeing here is activity has made the bulk modulus 0 in some pockets, and in those pockets you are seeing these singular features. So this is these two simulations differs in the like so this B tilde is going to 0 due to activity, and B3 is like producing these finite walls in the energy landscape. So without B3 you will hit a singularity, and this singularity are the like these are the one-dimensional version of the force chains we were talking about. We will see a 2D simulation in shortly. And with a small B3 term, B3 is like will give you a like length scale, a short length scale. So you won't hit the elastic singularity. You will stop just before hitting the singularity. And like we have calculated the approach to the singularity. Like you can like analyze those PDs I showed. And there are some exponents. So this is a finite time singularity. So at certain time t0, you will hit this singularity. So meaning that like starting from like if you switch on activity, you will eventually reach at what time you are going to get those stress fibers. So this sort of a physical measure of that time, and like there are some detailed calculations, and this is a large domain simulation. So you are getting many such, like these are all one-dimensional version of those stress fibers. And so you if you take a large domain, you first form those singularities, and they will merge and like they will coarsen. But the coarsening rate, it's a non-standard rate. So there is no university class like standard systems. And we have seen a very slow coarsening, logarithmic coarsening, in fact, and it depends on how long you are simulating, and
you don't have coexisting phases in this case. I mean, it seems you have a phase and then you have a high.
Yeah. So
right?
it is a coexistence of like low strain and high strain regime.
But the high strain is sort of sharp peak, right?
Yeah.
So it just keeps growing, I suppose.
Yeah.
So it's not. You don't have coexistence.
Yeah. Huh?
It's coarsening, but we don't know how.
Yeah, it's very slow, and the coarsening rate will depend on the turnover also. So I'll just show another video here. So here what we have done, we have taken a snapshot of this, and you have turned off the myosin unbinding binding, and then it coarsen regularly. So the coarsening of the singularities. So in usual phase segregation, you have domains, and the domains will coarsen. But this is like to study this mathematically is a bit of challenge because these are like coarsening of singularities. So these are very sharp peaks which will collide and coarsen. So
same thing happens in debating problems where you have a coexisting phase of one height and one at infinity as a matter of fact. The phase is at infinity.
Okay.
And it's going to keep growing the peak until it finally diverges.
Okay.
Yes, so we can discuss this later. Sorry.
Okay. So yeah. So Yeah, so this rate that you are seeing, that so these numbers, 0.5, 0.3, they really depend on my myosin unbinding binding dynamics. So they will change. Like these exponents will change if I take a different unbinding rate. So. Okay. No, I haven't. Mm-hmm.
You may think about the picture something like A plus A goes to A here. Your association
Okay.
every the particle A moves somehow, and then when you meet,
Okay.
they coalesce, and you will look at the density.
Okay, okay, okay. Another interesting feature is here you see a splitting. So here you will see a peak will split into two. Yeah, so a peak will split into two. So one peak, like this is some feature that I don't know how to explain this. So like we are still like sort of thinking about this, how to like to treat them as quasi-particles, or like we are all considering all that. Anyway, so yeah, so this is a 2D simulation. So this determinant, so this F is something called deformation gradient. So this is nonlinear elasticity now. So F is just the gradient of my displacement field, and I am plotting the determinant of it and its inverse. So this is what you are seeing is like the density distribution. So you get these sharp lines, and this is the trace of the stress tensor. And these black lines, what I have plotted, these black lines are the tangential stiffness modulus. So this is B, like bulk modulus plus shear modulus, but the bulk modulus is the tangent modulus now, like it's not about reference configuration. And this line marks where that B plus mu is equals to 0, in which regimes. So the point of this picture is the stress fibers that we are seeing here, they are marginally stable. So we are starting from an unstable system, and the system is tuning itself to marginal stability. So this is like we are thinking that this may be some new class of geometric self-organized criticality, where the order parameter is a tensor, and it coarses very slowly, and these lines, if you wait long enough, you will see that the whole system will be like will be filled up with these lines. So it's like the whole system will like these marginal lines, it will fill up the whole system. And of course there are certain regularizers for which it will stop somewhere, or like the rate is very slow. And so okay, so this is 2D, and in 3D, if are these movies, okay. So in 3D, you will see force sheets, like force chains, and this. So here we have plotted the highest strain, like where the strain is the highest, and this sort of looks like an active sponge phase, and like we are conjecturing that like these are minimal surfaces, like we have to test them, and also like you can like put this theory as a curvature flow. So you can write down the metric in the deformed configuration as my reference metric plus strain, and in the small strain limit at least, this metric evolution is governed by some. The second derivative of the metric tensor is some sort of curvature. So this is some sort of like not Ricci curvature, of course, but some sort of general curvature flow. So in Ricci curvature, flow, so this is like a result of the famous Perelman, that it will like the metric will become singular in some finite time. So we are trying to put all this work in terms of some generalized curvature flow, where you will hit a finite time singularity, like through activity. where the metric will degenerate on certain hypersurfaces, and those hypersurfaces, we think that they are minimal surfaces, meaning they have mean curvature 0, but we are still testing it. And yeah, so okay, so all that work was about like about certain phases of our phase diagram where you get like non-oscillatory solutions, and we have seen oscillatory solutions also, like standing wave, traveling wave, something called peristalsis, and another interesting solution where you start from oscillatory phase. So you start from sort of standing wave, it goes into a traveling wave, and it will move into something else. So it is slowly getting this modulatory like traveling waves, and it will eventually go into like a static solution. So this is like another route of getting those. So these are actually, if you put like this fat things, like it depends on the diffusion term in our equations. So this is another route of getting those stress fibers. So you are starting from an oscillatory regime in our phase diagram, but what you see is like oscillatory regime in terms of linear stability. So you expect oscillatory solutions, but you get a static solution. And this happens because, so this particular solution happens in a very like interesting regime where so this is something I have to mention here. So our the dynamical matrix of our system of equations is actually non-Hermitian, and it leads to something called exceptional point. So in usual stability analysis, so the way we usually like draw phase diagrams is based on eigenvalues, but in these non-Hermitian systems, the eigenvectors actually they are not orthogonal, so they can collapse on each other, and close to those regimes, your linear stability phase diagram is meaningless. So it won't give predicted like what you predict. And a crucial example of such exceptional points is usual fluid turbulence. So our Navier-Stokes equations, it is also like its linearization. The matrix is not Hermitian, so it has also exceptional points. And there is something in fluid dynamics called bypass transition to turbulence. So like it's pipe flow, and you are far away from, like your Reynolds number is very small, like from the critical, like very lower than the critical Reynolds number, but you still get into a turbulent regime. So that route is called bypass transition to turbulence, and that is related to this exceptional points. And in our system, like we have like really exceptional manifolds, like parameter, like surfaces on which eigenvectors are collapsing. So yeah, it's done. So we have done very anyway. So this is just a simulation in finite domain. It's not working, and like our objective is to get like a continuum theory of this kind of robots. So you start with a homogeneous system, and it will break up into these lines carrying tension and compression. So these are called tensegrity systems, tensegrity robots. And right now these robots are all static. Like you cannot change this bands and struts. So like our cell is like an evolving tensegrity robot. Like it's like you are making and breaking up these components. And so this is basically my end slide. So I thank Madan, and Lev was my previous postdoc supervisor, and Shoptorshi is like a PhD student at Simons. So he has done all the simulations that I presented, and like the whole work is summarized in these two papers that you can check out. So yeah, so this is my end slide. Thank you. Yeah.
We have time for one or two questions. Yes, please.
Just concluding, problem is with the length scales. So you don't, you approach them, but not like in nature they're approached. So apparently you don't release constraints effectively or whatsoever. You get trapped into some long-living metastable states. But how nature avoids them? What can be the answer?
One crucial thing that we have not, it's not in our theory, is that actin is, the material is also getting remodeled. So this is just one sort of first, it's like a very approximate theory of the actual stress fibers. You can, I am not saying that this is what happens inside a cell exactly, because there are there are many other factors, like where actin will polymerize and other facts. But this is sort of want some understanding, sort of like this theory of who was that? All biological systems are poised at criticality. That paper, William Bialek. So this is sort of our proposal that such materials maybe they can poised at some elastic criticality where you get these lines. So this is not exactly what happens in cells. I'm not saying that.
Might it be that catch bonds is not a good thing, that it's too a rigid constraint?
Okay.
And you have to make it not infinitely strong, the catch.
Okay.
Any other comments, questions? If not, let's thank Ayan again.
Thank you.
I guess that's the end of the day, and we meet at 9:30 tomorrow morning.
And we have dinner.
Dinner, of course,
Why, probably.